Okay, so we probably haven't started at the beginning of a microbiome labs presentation in a while. And some of you may not know that they have a vision statement, but they do. Their vision statement is resolving the global health crisis. Pretty lofty, eh? And we're getting there through our mission, which is making vibrant health accessible for all. So that's the mission that's working toward the vision. And our core values: innovation, service, elevation, integrity, and community. And this presentation is highlighting the spore differences. So we're going to discover what makes spores different than the reseeding method attempted by traditional probiotics. And this is just a colorful diagram illustrating that we are merely a collection of symbiotes or biots who form a holobiome, a superorganism. I don't know if you've ever looked at the human in this way, a new perspective. Symbiogenesis is the result of permanent coexistence of various biots to form the holobiont, namely the host and its microbiota. And that's by Richard Guerrero et al. The eukaryotic individual can be analyzed as co-evolved, tightly integrated prokaryotic communities. In this view, natural selection acts on the holobiont as if it were an integrated unit. Okay, so our new current understanding is through the Human Microbiome Project, and that article that we talked about in the previous talk was the resource for that technology, where we're really understanding that there's a lot more we don't know, that the human flora has never been completely characterized, limitations of the microbiology with obligate anaerobes. So some of the antiquated technology that other companies, probiotic companies, was based on very limited science. Now we're into the age of the metagenomics, and this allows for the characterization and study of the total GI microbial population as it relates to health, wellness, and disease. And you know, NIH launched this project. So this was back in 2015, and since then, we've got a lot more information to go on. So that what we've learned, of course, is there's a lot more organisms than they ever thought. So 1,000 different species, 100 times more cells than there are actually human cells of our body, and the bacterial genome outnumber the human genes more than 150 to 1. So we're really using a lot of their genes to perform our human functions, which is pretty amazing. No two individuals have the exact same composition, not even twins. At the phylum level, we see that there's, the phylum level is conserved, but the species level has a vast difference in different species populations. And these pop-ups, it's hard to read the last one. Okay, so here where do we get our microbiome? And this we focused on earlier, the whole seeding fallacy by probiotics, that seeding really occurs within 838 days. That creates your unique fingerprint that you'll have throughout life. Now you can influence that because antibiotics and lifestyle can greatly suppress certain keystone strains. And what we've tried to do with spore probiotics is to influence those keystone strains to grow and then protect the host, which is us. A diverse microbiome is a healthy microbiome, can do more functions, has more functional redundancies, which you'll find is important in regard to producing more metabolites, so that you don't run out if there is something that wipes out a certain population. Diverse communities are more resistant to invasion. And what affects the microbiome? Well, we know that age, diet, environment, antibiotic use, physiology, probiotics, and loosely genetics. And what we learned, really the current strategy of reseeding the gut with probiotics. Let's look at some of the fallacies behind that. First of all, let's look at the ingredients. We see in other, non-spore probiotics, you've got a lot of bifidobacteria, a lot of different types of lactobacillus, and in this one, let's see, bifidolactobacillus, streptococcus, thermophilus. And so we look at how are these probiotics prepared, in an air vat fermentation. So they're genetically adapted to the air. So will they even survive in the gut? And so what studies have been done to assure, are these companies doing SHIME studies, the simulated intestinal GI tract studies that really would show what's going on in the gut. And we find out after, this is a particular study that was done, that only 4 out of 35 strains even survived to enter the large intestine. And the survivors, if they did get to the large intestine, only 50% of them survived. So now we're looking at 2. And then if they don't study, how they react with the environment, they might even be causing more inflammation. So that is a problem. This is a study, the Silker Inc., which is a food microbiology. They do survival of probiotics in simulated gastric fluid, so kind of a simulated type of SHIME. And they studied, yogurts, and both yogurts saw 99% of the probiotics die in the stomach. The leading supplement brand had nearly a full-fold overage, but over 99% of them died in the stomach. And it was only the Megaspore that showed 100% survival with no loss at all. So there are studies, that back up, what we're saying about the spores. So what are other issues in regard to that fallacy of seeding with probiotics? Here's a big one, the aerobic bacteria seeding in an anaerobic environment. Do they fit in the gut and the binding sites for the gut? So, here we've got a study that was done in 2009, a comparative study on adhesion and recovery of potential probiotic strains of lactobacillus. And they showed that at best, the adhesion was 38% and was 20%, the rest went down from there. So we have to say, is this natural? Does it really occur in nature? Are we, did our ancestors, ferment lactobacillus and, take it, as a supplement? Well, of course, no. And so it really doesn't fit the environment, and it wasn't, natural to the environment. So does it happen in nature? No. The issue of finite overload goes against expanded diversity. Quality. Some strains used may not have studies to show their benefit. Mislabeling of probiotics has been found to be a huge issue. So several documented cases of mislabeled probiotics. A panel of the EU experts in microbial characterization found the following. Probiotics were incorrectly labeled with the wrong strains. Products contained markedly reduced numbers that they contained, and found presence of strains not labeled. In some cases, they were pathogenic. So the quality control of probiotics is lacking, according to this study. And after 16 probiotic products available in local California stores and also online, they found only one of the products exactly matched the bifido species claim on the label. Some products had pill-to-pill and lot-to-lot variation. So there's a problem within the industry itself. So the reseeding plan of using non-spore probiotics failed at the survival at the fitness, at the natural strategies, and at the quality. So what is the spore probiotic supplementation? Okay, so we're not trying to reseed. Spores are transient organisms that are found in nature. So we're back to, yes, they fit the gut. They have been part of the human gastrointestinal tract since the dawn of time. It's important for them to have exposure, to be exposed to them as early as possible. Studies show that infants that are not exposed to these spores will develop autoimmune and immune deficiencies. And we know this, from studies of indigenous populations. Here's one. We know that human gut microbiota community structures in urban and rural populations in Russia, the human gut microbiota community structures in urban and in populations in Bangladesh. So we know what a strong microbiome should look like. And this is a quote here that, what is nature's design for supplemental probiotics? This quote is, the Chuck Norris of probiotics, the tough guys, has to do with bacterial spores. So in particular, bacillus spores, as they are the most widely studied and most widely used probiotics outside of the supplement market. Bacillus spores, that means that in Europe for the last 60 years, they have been commercial products, pharmaceutical drugs, since 1958, used extensively in agriculture and aquaculture. Most widely used and well-studied strains in humans are the Bacillus subtilis, lichen formis, the coagulans, the Clausii, and the indicus. Now, I go back to this agricultural and aquaculture. It always seems to be the veterinarians, when it comes to nutrition, are always, decades ahead of human medicine. And it's always been that way. So, key features of bacterial spores. First of all, they form a robust endospore, which can withstand the harsh temperatures, desiccation, low pH, the gastric barriers, antibiotics, UV radiation, solvents, enzymes, and even high pressures. They've found spores in archaeological digs that are thousands of years old and can still germinate. They are found all over the environment: soil, vegetation, dust, rocks, aqua environments, digestive systems of insects, marine life, and mammals. Spores remain dormant for over 50 million years. Well, that depends on your perspective. Can't really prove that, can we? Found all over the ancient environment, ice core studies. They colonize very effectively in the human gastrointestinal tract and have been found to colonize very effectively in the gut of several different animals, are found as part of the normal human commensal flora, are considered keystone strains, long history of use in industries where efficacy is closely measured, like the pharma, agriculture, aquaculture, extremely safe, and are considered true commensal organisms. So the most important function of a true probiotic is to protect the host. Okay, and that's where, again, even if a Lactobacillus bifidus probiotic shows a limited function because of their digested protein quorum sensing ability, they still are limited to one strain, one condition, where you're looking at spores, which through the SHIME studies are shown to have over 32 beneficial features for the human gut. So I'm going to end this presentation and see if there are any questions. So ask away. Oh, sorry, let me make sure that the mute is off. Okay, now mute is off. Mute off. Did anyone have any questions on the the seeding or the dispersal for setting up the mature microbiome, or the second presentation, which was the difference between spores and regular non-spore probiotics.
Thank you, Tracy. This is really helpful.
Oh, super.